Hello dear students, I am Dr. Jisha Krishnan from MES KVM College Pharyngeal. Here I would like to share some video lessons especially for third semester BSc Zoology students of your core paper Zoology, and I am starting with the first module, Introduction of Phylum Chordata, and today I am going to talk about the topic, the general, fundamental and advanced characters of phylum Chordata. So let's move. Have you heard about this phylum Chordata? It represents the last phylum of kingdom Animalia. If you are tracing back to their ancestry, you can say that their ancestors were known to be appeared in the Cambrian period over 530 million years ago. The word Chordata comes from two Greek words, chorde and eta. Chorde means string or cord and eta means bearing. So the members are popularly known as cord bearing animals. Okay. And in which type of organisms are generally found in this particular phylum? They generally involve fishes, amphibians, reptiles, birds and mammals. Around 64,832 species are known to be existing on record, and out of which they are categorized under just like this much number, 32,120 in fishes, 6,771 in amphibians, like that, and from which one is smallest and which one is largest. The smallest one is called the Philippine goldie, who is having 10 millimeter in size, and the largest is, we know that, blue whale. It is having 35 meter in length. Then comes what is the general characters of this particular phylum. First one is their habitat. In which type of habitat you can see they are aquatic, terrestrial as well as aerial. In these three habitat you can see this chordates. Then most of them are free living. No parasitic forms are found. We know that most of the parasites are found in the invertebrate phylum like Archaeominthes as well as which one? Platyhelminthes. Okay. Then they have bilateral symmetry. I think there is no need of much more explanation. Then they possess a post-anal tail. Then they possess exoskeleton. Exoskeleton means the external covering which provides protection to our body. For example, in the case of mammals, we have skin. In the case of birds, they have feathers. In the case of reptiles, they have scales, etc. Then they are triploblastic, which means their body wall consists of ectoderm, mesoderm and endoderm. Then a true coelom, a true body cavity that you can see in between alimentary canal and outer body wall. Then comes their endoskeleton. What about their endoskeleton? Endoskeleton is nothing but these are cartilaginous or bony structure which is generally providing support and shape to our body. Okay. Then they have a complete digestive system. The digestive system starting from mouth leads to pharynx, esophagus, stomach, intestine, rectum, anus. So in this manner a complete system is formed, digestive system is formed. Then as far as the blood vascular system is concerned, they have a well-developed blood vascular system which is closed with a true heart and associated blood vessels like what are? Artery, venules and capillaries. Then comes the excretory system. They have a very good excretory system consisting of either proto, meso or metanephric kidneys. Then the nerve cord is composed of a dorsal tubular one. Actually this is the brain as well as spinal cord. The dorsal tubular nerve cord consists of the brain and spinal cord of our body. Then they are disexual, which means sexes are separated. Male as well as female individual can be seen. Then what is the fundamental characters of chordate, or what are the characters which is unique to this particular phylum? Their unique characters are actually as follows. They possess a notochord, then they have a dorsal tubular nerve cord and they have pharyngeal gill slits. Here you can see that you can clearly understood by seeing this diagram. This yellow colored one is called the notochord, and this is called the dorsal tubular nerve cord, and this is called the pharyngeal gill slits. Those organisms which possess all these three characters can be treated as a chordata. Then let's move to the detail. What is meant by notochord? Notochord is also called chorda dorsals. Here you can see that this yellow colored one is the notochord. And actually it is a part of axial supporting endoskeleton, which means I told you that endoskeleton is a structure which provides shape and support. Okay, so this will provide support to our body. Okay, this is a function. Then actually it is an elastic. What is the nature? It is elastic, cylindrical and rod-like. And where it is positioned, it is found just above the alimentary canal. This is alimentary canal, just above the alimentary canal or just below the dorsal tubular nerve cord. This is a dorsal tubular nerve cord, this is the alimentary canal. In between these two, you can see the notochord. Okay, then this particular notochord is made up of a particular tissue that is called notochordal tissue. So if you are taking a cross-section view, you can see that they composed of an inner notochordal tissue, and just above this notochordal tissue you can see a protective covering that is called a notochordal sheath. Okay, so come to the notochordal tissue. Notochordal tissue composed of two distinct regions, an inner vacuolated region and an peripheral non-vacuolated fluid-filled region. What is the peculiarity of this region is that mainly this notochordal region, this is called notochordal region and notochordal cells. This fluid-filled peripheral region is called the notochordal cell. It will provide or it will secrete some fluid into here, and this fluid will create some pressure, and this pressure generally provides a stiffness to the notochord. Okay, then comes the notochordal sheath. Just above this notochordal tissue you can see the protective covering that is called a notochordal sheath. This region is also divisible into two distinct regions, an outer region that is a primary sheath which is elastic in nature, and an inner secondary sheath which is somewhat fibrous in nature. So that's all about the histology of notochord. And from which layer it is formed? It is truly endodermal in origin. Then persistent. How they are persisting in the body. In the case of protochordates, that is cephalachordate as well as the urochordate, that is protochordates are also called lower chordates, they are persisting in differently. In the case of cephalachordate chordata or amphioxus, this is found throughout their life. While in the case of ascidians or urochordate, this is generally found in the larval stage. Okay, which means as this urochordata becomes adult, we cannot see that particular structure. While the case of higher chordate or vertebrates, they persist only in the embryonic stage, which means that when they become adult, this particular structure is getting replaced by another structure that is called a vertebral column. Okay, so that's all about the notochord. Then comes dorsal tubular nerve cord. This is the dorsal tubular nerve cord. Where it is found just above the notochord, and it is in hollow, unpaired, non-ganglionated cells. And this tubular structure encloses a canal that is called neurocele, and they are strictly ectodermal in origin. Okay, so from which layer? From ectoderm, this dorsal tubular nerve cord is formed. Then actually this structure gets anteriorly differentiated into brain and posteriorly gets differentiated into spinal cord. So brain and spinal cord constitute what? Dorsal tubular nerve cord. And this brain and spinal cord is filled with what? Cerebrospinal fluid. By the analysis of this dorsal tubular nerve cord, we can predict some kind of some aspects in evolution, or we can reflect ancestry, stating that the presence of this dorsal tubular nerve cord in the dorsal region of chordates might be indicating that their ancestors might be a free-swimming pelagiform, which means the ancestors of phylum Chordates are found in the surface of water. Okay, so this is the reflection of their ancestry. Then comes pharyngeal gill slits. This is another unique feature found in this particular phylum. Here you can see that these openings are called pharyngeal gill slits, and hence they are also called a visceral cleft. These are paired lateral opening on pharyngeal wall. Here you can see that on the wall of the pharynx, you can see this structure. Here they are found in this region. The right side it is found in quite opposite to the left side. On the ventral side, you can see that they're paired structures. Here, if you are concentrating this particular slit, its pair will be found on this opposite side. Here also its pair will be seen on this opposite side. Okay, so these are paired lateral openings, and the main function is mainly concerned with the outlet of respiratory current. Here you can see that through the mouth water enters, and through after respiratory exchange it will move out to this opening. Okay? So it acts as an outlet of respiratory water current. And how they are persisted. In the case of protochordate like cephalochordate and urochordate, fishes and cyclostomes, they are found throughout their life. Okay, while the case of amphibians, it is found only in the larval stage. You know that tadpoles are actually aquatic forms, so only in the larval stage you can see this amphibian larval stage only you can see this pharyngeal gill slits. And in the case of terrestrial vertebrates, that is adult amphibians, reptiles, birds and mammals, this structure found only in the embryonic stage, which means that when all these organisms become adult, it gets replaced by the terrestrial air breathing organ, that is lungs. And if we are considering the number of these gill slits, it varies in different organs or different groups. Actually, when advancement increases, the number of gill slits get decreases, which means that a higher number of gill slits is found in lower chordate, while higher chordate contains lesser number of gill slits. And they are generally developed by means of the outpushing of pharyngeal wall, and this all gill slits will meet with the surface of ectoderm and forms the pharyngeal roots or pharyngeal pouch. How they are formed? This pharyngeal pouch is formed by the union of this pharyngeal gill slits with the outer surface of ectoderm. Okay, this is found on the pharyngeal wall. Pharynx is found on this is found on the inner region of our body. So it will be meeting, or it will be communicated, it will be united together with the outer surface of our ectoderm and forms this structure. In primitive chordate as well as fishes, you can see that this pharyngeal pouches is lined with the gills. Okay. Then comes what are the advanced characters of chordate. Actually, most of the advanced characters are seen in relation with, or you can most of the characters show a shared character with the invertebrates. Okay, the first one is called axial organization with bilateral symmetry. You know, I think you know what is mean by bilateral symmetry. When a clear cut is made in the correct anterior posterior direction, you will get equal halves. Here it is a mammal, you will get equal halves, and when a correct here it is an frog, when a correct plane is made here through this anterior posterior direction, you will get equal halves. So their body generally possesses two different kinds of axes, that is an anterior axis and a posterior axis, and also a dorsal surface and a ventral surface. Then they are triploblastic, no need of explanation. Body wall consisting of ectoderm, mesoderm and endoderm. Then they are true coelomat, a correct body cavity that you can see in between alimentary canal and outer body. Then metamerism, that means a segment organization is generally found in metamerism is a condition, and this is generally in higher chordates. So this metamerism is generally found in the primitive stage or embryonic stage, gets replaced in the adult stage. Then tube within a tubular, which means alimentary canal which is provided with a two opening, that is an outer mouth and our anterior mouth and a posterior anus like structure that you can see here in this organism. So that's all about the advanced chordate characters. I hope you understood.